She played a leading role in showing that specialization of brain regions extends to such abstract concepts. She also has recent, exciting work examining the development of such regions in infants, as reflected in her title, Infant Brains Developing in a Social World.
Rebecca. Thank you. Thanks so much for having me. This is a really exciting day to be a part of. And I was actually excited to hear Phil Sharp say that in the very beginning, the vision that Laurie and Pat had for the McGovern Institute was understanding the biological basis of the higher functions of the human brain, with applications not only in health, but in understanding society and pedagogy and the great, unique accomplishments of human intelligence. That's exciting to me because that's, I think, the vision that guides my own research. And in particular, my view is that in order to understand the remarkable accomplishments of the human brain, we have to, among other things, understand development. We have to understand how children and children's brains develop and learn and acquire these functions, in particular from social contexts. If we think about what are the truly unique capacities of humans for language, for communication, for cooperation, for teaching, all of this arises in and is learned from a social context. And in fact, from hours after they're born, human infants are not passive recipients of their environments. They have active preferences. They choose what to look at and what to learn from. And they actively seek specifically social experiences and preferentially learn from those experiences in the first hours and days of their lives. So in my lab right now, we've been studying that process, both from a cognitive perspective and from a neural perspective. I'm trying to use neuroimaging, non-invasive neuroimaging, to directly study the brain regions that are involved in infants seeking and processing social experiences. And so I'm going to tell you a little bit about that work today and some of the surprising conclusions it suggests, both for understanding infants and for understanding brain development. Okay, so we've known for more than a century that infants' brains are anatomically dramatically different from adult brains. You can just look at this picture. The most obvious difference beyond the size is the color, right? So on the left is a typical adult brain. On the right is a 4-month-old infant. And you can see that the white matter, the thick white myelinated tracts that are so clearly visible in the adults' brains, are evidently dramatically not present in the babies. The timeline of myelination, by which tracts become myelinated in infants, is incredibly slow. It's slower relative to our nearest primate relatives, even than cortical development. And it's very exaggerated, especially in higher order functions like prefrontal cortex that subserve social function. And so because what people have known about infant brains for 100 years is that they are dramatically different from adults in this anatomical way, they're also remarkably resilient to injury, we all expected that the function of cortical regions would also be dramatically different in infants, that the basic divisions of labor, what the functional roles of different cortical regions that we had learned in adults, that this would be dramatically different if we could study babies. But we also thought that it wasn't possible to study babies, and that's what's changed in the last 5 years. It's become much more possible to ask these questions by directly neuroimaging infant brains. So to test these ideas directly, the first thing we did is measure infants' functional responses to faces. That's because pre-verbal infants prefer to look at faces. So this is an eye tracker experiment we ran a few years ago, and each dot is a single infant. You can see where they're looking in these movies. You can tell that even in these relatively complicated naturalistic scenes, infants systematically prefer to look at faces. And in human adults, we know that there are multiple patches of cortex that play specific roles in recognizing and understanding faces. And these cortical regions are necessary for typical adult face perception. So the first question we want to ask is any part of this cortical organization present in infants? To do that, we had to do fMRI of infants, and so we commissioned infant-sized coil for the Siemens MRI machine, which is right downstairs. And we scanned human infants aged 3 to 8 months. They're scanned awake because we need to see them looking at their social world with an adult in the bore with them to monitor their well-being and alertness. And it's through the mirror that's placed over their heads the infants can see videos playing on the screen at the end of the bore. The videos show dynamic, interactive, smiling faces and naturalistic scenes, the kind of thing they might see in their neighborhoods. So through this mirror, the baby sees the film. And the most difficult part of this experiment is that MRI signals are highly sensitive to motion. We can't see an image if the baby is moving even a millimeter. And we can't put pressure on an infant's soft skull. So the actually hardest part of this experiment is we just have to wait for them to choose to lie still to watch our movies. Okay, so as I said, the first big picture question is, would any of the face responsive patches of cortex in adults already show any preference for faces in infants? So show greater hemodynamic activity while watching the faces than the scenes movies. I'm showing you two such regions here in the adult, which are the FFA, fusiform face area, which was discovered by Nancy Canwisher, and a region in the superior temporal sulcus involved in perception of face motion in adults. And here's what we find in a 6-month-old infant, is the exact same regions already respond more to movies of faces than scenes. And looking at this more quantitatively, we can use some parts of the data to find these regions and separate data to quantitatively measure their responses. And what we found is that in every cortical region, that in adults there's face selective responses, in infants there's already a significant face preference. You can see that the hemodynamic, or the bold response in infants, is smaller, and the difference between conditions is also smaller in babies, but there was a clear and significant preference for face movies in each region. And this is true of the fusiform face area and the STS I'm showing here, but one brain region in particular caught my attention, even beyond these ones, which is we saw the same pattern in medial prefrontal cortex. That's a patch of cortex right behind the middle of your forehead. So of all the regions where we saw face preferences in infants, this one was the most surprising to me for a lot of reasons. The first one is that, as I told you, myelination reaches prefrontal cortex the latest of anywhere in the human brain. And this is literally the last place I would have expected to see adult-like functional organization in a human brain, in an infant's brain. This is a picture of the myelination, a direct measure of myelination in an infant. And by 5 months, which you see in the latest plot here, there's really almost no myelination of the tracts leading into prefrontal cortex. Okay, so that's the first reason. The second thing is because of what the function of these brain regions is in adults. So the fusiform face area, as I said, is in recognizing faces. The STS in recognizing face motion. Those are high-level perceptual regions. But the medial prefrontal cortex is not a perceptual region. In adults, this brain region plays a high-order and abstract role in processing social information, particularly in representing information that is self-relevant or valuable to the perceiver. So what I'm showing you above, these are the results of a meta-analysis just published this year, which included thousands of fMRI studies in human adults and showed really strong association of these medial prefrontal regions with higher-order social perception, specifically with respect to the self and what is valuable to the perceiver. So is it possible that in pre-verbal infants, medial prefrontal cortex already plays a role in processing self-relevant and valued social information? I think it does, and I think that has important consequences, both for learning in typical infants and for understanding developmental disorders. And I'm going to show you how we're continuing to test this idea using a different neuroimaging technique, one that's a little easier for babies, called near-infrared spectroscopy. This is what it looks like to put a cap on a baby for near-infrared spectroscopy. This, like MRI, this measures blood oxygenation in a baby's cortex, but here using a lightweight cap so the baby can sit on the parent's lap while watching movies or playing with toys. Okay. So that cap is not measuring medial prefrontal cortex, but this one is. So this is a cap that we designed to measure hemodynamic responses covering most of prefrontal cortex, including both medial and lateral regions. And I'm going to start by telling you what we found in medial prefrontal cortex, and then I'll zoom out to the bigger picture. Okay, so babies in this experiment are wearing a cap measuring blood oxygenation responses throughout their prefrontal cortex, and we give them a chance to look at and learn from 2 different people shown in videos. And the first one speaks in what's called infant-directed speech, which is a kind of prosody and valence that babies particularly like. This is what it looks like.
Gesi fawo namu, mufa nasi gewo.
Okay, so this highly exaggerated pitch contour called infant-directed speech is the kind of speech that babies prefer to listen to, and actually we found later in these experiments that they learn best from. Okay, the other speaker speaks the same kind of nonsense but in neutral adult-directed speech.
Ti la bu. Bu do la pa ku ti.
Okay, so we hypothesized that infants might perceive the infant-directed speaker as more self-relevant or socially rewarding than the adult-directed speaker, and so might engage medial prefrontal cortex differentially, even though both videos show a face of a person talking to them. And that's what we found. So this is the first set of data from a group of 9-month-old infants, where the blue is hemodynamic response recorded during the infant-directed speaker, the green is during the adult-directed speaker, and we've since replicated that in a group of 6- and 7-month-old infants, showing the same pattern. We have another replication still going on. Okay, so that shows that medial prefrontal cortex is more active during the infant-directed than the adult-directed speaker, but that's actually not what I'm most excited about in this experiment. Here's what I'm excited about. After this initial exposure, we then gave babies a preference test where they saw both speakers silently smiling at them. And the first thing is, infants choose to look at the speaker who previously was infant-directed. So having learned that one of these two speakers speaks in a way that's self-relevant or socially rewarding, now babies choose to keep looking at that speaker even though she's not talking anymore. Okay. But this is highly variable across babies. And so what I'm going to show you here is each individual baby out of the almost 50 babies in this experiment. On the y-axis is how much their medial prefrontal cortex responded during the initial phase when they were listening to the speakers. So on the x-axis. On the y-axis is their preference test, how much they preferred to look at that speaker. So you can see that we can explain a lot of the variance in the individual babies' social preferences from the two silent speakers by how much their medial prefrontal cortex responded at the beginning of the experiment. The more your medial prefrontal cortex responded to the friendly person in the first phase, the more that baby will choose to look at that person later on. So that's already pretty cool, because we're using, not only is medial prefrontal cortex active for a stimulus that's socially rewarding or self-relevant to the baby, but how active it is plays a direct functional role in predicting the baby's later behavior. Right? That's behavior measured after the brain. Okay, but here's why I'm really excited about this, is that I just showed you one piece of a much bigger experimental paradigm. So I showed you that we hypothesized that medial prefrontal cortex is involved in processing self-relevant or socially valued stimuli. We tested this with two speakers where one of them talks in this exaggerated prosody pattern, and we tested babies' preference for which speaker they looked at when they were silent. So babies were randomly assigned either to be in this condition or to be in a different condition. In the other condition, we've varied something different. We varied the statistical structure of the speech that the speaker was saying. So one speaker spoke in a statistically structured speech where if you hear mo, you know you hear foo. If you hear gay, you know you hear na next. So it's nonsense, but with statistical structure. The other speaker spoke sequences of syllables that were not predictable. So mo, 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 gay, 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 fa, 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 fa. And afterwards, we gave babies a chance to look at two kinds of syllables that fit the pattern they had learned, or syllables that violated the pattern that they had learned. So again, we know from lots of past behavioral research that if babies have learned the statistical structure in the first phase, now they'll prefer to look at the novel stimulus, the one that violates their learned expectations. So now we have two different behaviors that babies do by the time they're 6 months old. One is they prefer to look at people who spoke to them in infant-directed speech. The other one is they prefer to look at sound streams that violate their expectations for statistics. And the key hypothesis here is that these two behaviors that babies do might come from different neural systems in babies as they do in adults, and be associated with different compartments within prefrontal cortex. So in adults, learning statistical structures and guiding attention to violations is associated with activity in lateral prefrontal cortex. And this predicts a double dissociation in infant prefrontal cortex, two different prefrontal cortical regions predicting two different behaviors in response to the very similar stimuli, a person talking nonsense in a video. And that is what we found. So this is the first experiment I showed you where babies got the high prosody, low prosody, and were asked who they wanted to look at. And as I showed you, this is now in the 6-month-old babies only. The left medial prefrontal cortex activity during learning predicts how much they look at that speaker during test. There was no such association in lateral prefrontal cortex. By contrast, if we manipulated the statistical structure of the speech, then activity in medial prefrontal cortex did not predict looking afterwards to the violation of their learned structure, but activity in lateral prefrontal cortex does predict it. Okay, so we found a double dissociation between two regions of prefrontal cortex in 6 and 7-month-olds, exactly as would be predicted from the functions of these regions in adults. Okay. So I think this is pretty exciting for a whole bunch of reasons. The first one is that despite the dramatic anatomical immaturity that I showed you at the beginning, infant cortex is spatially and functionally organized, even not just in perceptual regions, but even up to and including abstract high-level functional regions, and even including prefrontal cortex. I think this raises a whole bunch of interesting questions. One of them is, what's myelination for, right? If babies' brains brains are functionally organized just like adults, if the same prefrontal regions are doing very similar jobs in the very first months of life, what changes when you have this dramatic change in the myelination of your cortex? That's only one of many things I think is interesting here. The second thing, and this is more about infants, is typical cortical development is influenced both by the social information that infants are processing, but also by their endogenous social motivation, their choice, their preference of who to look at and what to learn from. So very young infants, and this has been measured down to 36 hours post-birth, preferentially look at and learn from socially valued sources. And that is imbued in the function of cortical regions. So early cortical regions functions are not just perceptual, not just responsive to the environment that the baby gets, but actually actively guiding infants in what they choose to learn from. And finally, connected to Bob's articulation that we're not just trying to understand the brain, we hope eventually to help people. I think the capacity to make sensitive behavioral and neural measures of infant social development is really exciting for lots of reasons, but one of them is to have a baseline for potentially measuring altered social function in developmental disorders. Okay. So with that, thank you so much to Pat, to Lori, and to the McGovern Institute for many years of support.
Thank you, Dr. Saxe. And do we have questions?
Rebecca, this is phenomenal work, and that picture is just mind-altering, I have to say. So I have first a comment and then a question. So watching my 6, 4, and 2-year-olds learn, I'm realizing just how much more emotional spillover there is in the younger they are, and how much that spillover is reduced. I'm curious as to whether that spillover has anything to do with myelination. In other words, making sure that these neurons are not just affecting everything in the brain. This is the first question. And then the second question was whether your video for adult-directed versus child-directed, did you try that on adults as well? Because as an adult, I actually responded to the speaker on the left more strongly than the speaker on the right.
Yeah. So those great questions. So the first thing is, we all assume myelination affects precision, right? Affects efficiency. So timing, precision, and efficiency. That the messages you intend arrive, and they arrive when they're intended to arrive. And of course, myelination is not the only thing that happens in infant brain development. It's just the thing that's easiest to see on a scan, right? So there's also lots of pruning of individual synapses happening at the same time that we can't see. So all kinds of changes happening in the brain. I think there's lots of reason to think, as you're saying, that what should happen is a narrowing of the information transmission between regions, where intended information gets transmitted and not intended information doesn't get transmitted. And so all of that seems very sensible at a word model level, right? And in every worked example where in animal models people have been able to study it, that seems like about the right example, or about the right way to think about these anatomical changes. I am just struck by how similar these functions manage to be despite such a dramatic loss of, or such a dramatic difference in the scale of myelination. I would have thought by the time you're getting all the way to prefrontal cortex, that's very multisynaptic, right? You've had to go through many different tracts to get all the way there with the information. It is surprising to me how little has been lost, in the sense that we can still see all these major functional divisions. Having said that, babies are not good at that many things, right? And so one of the interesting ideas that I've been thinking about is, it's been known for a long time, but most famously, Chomsky articulated a difference in infancy between competence and performance. That is, babies seem to be able to learn an incredible range of things that they are not able to express in their behavior. The most obvious example of this is language, where babies' understanding of language outstrips their ability to produce language by many orders of magnitude in infancy, right? Infants can understand complicated sentences when they can produce one word, right? So that's the competence-performance distinction. And then there's examples of that in every area of cognition, where babies' understanding of the world around them seems to vastly outstrip their ability to act on it. So I think it's interesting to wonder whether learning is much less sensitive to precision and timing than acting. And so maybe babies are accumulating an understanding of a world long before they can use that to act on it. That's one idea.
Oh, there's a question here.
Thank you. This is fascinating and wonderful information that you're sharing with us. When you say that babies look at and learn from socially valued sources, what are you defining there?
Such a great question. So there's an extensive literature characterizing what babies prefer to look at. And when I say socially valued, that's a hypothesis, right? I'm hypothesizing that what the common function there is something that babies value. It's actually also self-relevant. So for example, babies prefer to look at, they prefer to look at a human face to almost anything else, especially right when they're born. Very soon after that, it's not actually whether it's a human face or not, but whether it's something that has responded to them in a contingent fashion. So babies prefer something, it could be a person, or actually can be like a blob, if its behavior depends on the baby's behavior. This, a famous example of this is called the still face paradigm. If you put babies in a face-to-face interaction through the old school version of Skype, right? So direct video link, let babies interact with somebody for a while, and then you switch to video feed, a loop. So the person is doing exactly the same behavior, the same positive voice, the same child-directed speech, but it's not reacting to the baby anymore. Babies detect that very quickly. By 3 months old, they can tell the difference, and they don't like it. In fact, they cry, right? So even though it's still a happy, nice person reacting to them, they will not respond to that stimulus. Actually, there's a neat twist on this recently that, on the other hand, so Pat Kuhl was just here last week, and she's very famous for showing that babies cannot learn language from a video. Well, what has recently been discovered is they can't learn language from a video, but if you, before the video, start with just a brief Skype session, so that for a little bit they think they're in a real interaction with somebody, and then there's that same video that before they couldn't learn from, now they will learn from it. So they'll extend the assumption that this is relevant to me for a little bit of the video. Now, at some point, they're going to detect it's not a video. I mean, it's not live, it's a video, and not like it. But you get this little window where if they think that person's really talking to me, that they will learn from the same video they previously would have learned from. Okay, so what do babies prefer to look at? They prefer to look at somebody who has reacted to them. They prefer direct gaze to averted gaze. They prefer somebody who calls their name to somebody who doesn't call their name. They prefer somebody who uses infant-directed prosody to somebody who doesn't use infant-directed prosody. And they will also prefer to look at somebody who previously touched them in gentle touch or rocked them. Okay, what is that set of things? It's caregiving behavior, human-adult caregiving behavior, right? Direct gaze, calling the baby's name, looking at them, using infant-directed speech and infant-directed touch. So babies very early on recognize those behaviors, prefer them while they're happening, remember who did them, and prefer that person later. And then in our own studies, we've shown they also learn the speech of that person better. So even though they've heard exactly the same amount of speech from these two speakers, they learn the speech better from the person who is acting as if it was relevant to them. Now, we say, okay, let's call that socially valued, but that's a hypothesis, that that's the right way to describe it. I will say So I am a neuroscientist, and the fact that it comes down to the same brain regions that in adults also have been described as socially relevant. Not that we know better what it is in adults, right? But again, the same things reward for the self, social reward for the self, self-reflection about me. In particular, those medial prefrontal regions, one of the things they like doing best, if you want to play this game, is thinking about the traits in myself I most value. What is it I most would like to be? I would like to see you to see me as a competent and warm leader. And as I think of myself that way, my medial prefrontal cortex goes crazy. Okay, so that task of thinking of what I most aspire to be like, that's what Western adults like to do with their medial prefrontal cortex. What babies like to do with their medial prefrontal cortex is have somebody talk to them in a warm voice. Tell them things with this high prosody. Okay, is that socially valued? I don't know, but it's something very close to that.
We have another question over here.
Hi. Genetically based developmental disorders contrasted with environmentally based developmental disorders, particularly when observations in some families where the baby, the moment it is born, is exposed to intonations in voice which are not endearing. Sharp contract associated with or without physical abuse. Any thoughts on that? Because that's a fundamental thing in terms of developmental disorders that are environmentally induced.
Yeah. So I think these questions are really, really hard. I will say that, so even just talking about infant-directed speech, this prosodic contour, this claim that babies learn from infant-directed speech better, psychologists have been making it for a long time, for 50 years almost, in various ways, and very much for the last 30 years. Anthropologists have been very critical of that claim because this specific behavior of being one-on-one with a baby, looking face-to-face at them, and then talking to them in that high prosodic contoured speech, that varies across human cultures in the degree to which that's a normal thing to do. In our culture, we're supposed to do it all the time. In some cultures, that's a really strange thing to do, and you shouldn't do it very much. There's cultures that encourage you to carry a baby, but not particularly talk to it for months or even up to a year. And anthropologists have accused psychologists of studying only weird babies, Western educated, industrialized, rich, and democratic, right? Only the babies who come from our particular little world, and to treat as neurologically normative something which is actually just culturally normative. And so now almost everybody who works on this infant-directed speech preference in babies and what it has to do with, for example, it predicts later language learning in our culture. Like, what does that mean thinking about a broader array of parental behaviors, and how sensitive are babies to variability both across cultures and then across families, right? So the question you asked is, what about other kinds of parenting even within our culture? I think there's a huge amount still to be learned and understood about, for example, babies' tolerance to really different rearing strategies. And in every example where this has been studied well, it seems like human infants are very tolerant to a very wide array of different rearing strategies, right? So that even cultures in which it is normative barely to talk to babies at all, those babies do learn to speak, right? They become competent adults in their culture. So that says that maybe babies are very resilient across a wide array of normal behaviors. I think we don't even really understand how that works. And yet, babies are sensitive to whether they're being neglected, right? And social neglect is incredibly bad for babies. And the powerful research in the orphanage studies by Chuck Nelson shows that you can neglect a baby too much, right? You can really harm infant development through extreme neglect, even without any abuse. And so, I think that these questions about environment, also genetics. One thing that I'm very influenced by is the behavioral genetics research. They use genetically informed designs to try to sort out how much of the variance in infant development comes from genetic variability and how much comes from family environment. I'm convinced by that research that at least typical variation among typical families is not as predictive of most measures of infant development as genetic variability. So that is what the answer you get from genetically informed design. Is that among healthy, wealthy, well-educated, typical participants in psychological research, most variability comes from genes.
Thank you.
Okay, thank you for your questions.